Hello, everyone. So we're just going to give everybody a couple minutes to get connected. Heather, are you here? So we're still waiting. Oh, you are? Okay, sorry, I couldn't find you.
I'm here. We are recording, and I think everybody should still be able to see the link. If anybody is interested in reviewing the posters, that could be students, TEPIs, family members, anybody that would like to add a review is welcome to go to that review form and add some comments or feedback for those that are presenting their work today.
Excellent. First up is going to be Chloe. Chloe, are you with us? Can you unmute yourself, Chloe?
Yeah, hi.
Hi, Chloe, great. So if you would like to introduce yourself, tell us a little bit about yourself, and then go ahead and just as a reminder, you have 5 minutes for your presentation. If you are down to the 1-minute warning, I'll make a normal announcement just to say, hey, 1 minute left. Those of you that have questions, you can type your question into the chat feature. You can also raise your hand and be unmuted and ask questions, but we do try to keep the presentations to about 5 minutes. Okay, Chloe, the time is yours.
Okay. I'm Chloe. I'm a 9th grader at American Heritage, and I did research on using anti-SMASH to compare the antimicrobial genes of commensal E. coli to pathogenic E. coli. Next slide, please. So antibiotics is arguably one of the greatest discoveries of all time, but with this discovery came a very big problem, and it is that of antibiotic resistance. This could be caused by 2 things. Number 1 being the indiscriminate prescription of antibiotics, and 2, when patients would not finish their full dosages of their antibiotics. A tangential problem of antibiotic resistance is that of superbacteria. One you may heard of is MRSA. Now, after this problem started to become more and more prevalent over the years, the medical community started to put some research into it. And we actually found that the very thing we're trying to kill can help us create a new antibiotic. And there are 2 main ways I found that this could happen. The first way being the way that the Tiny Earth Initiative did it by using soil bacteria, or the University of Germany used bacteria that's already found in humans to do so. Now, there are 2 different types of bacteria that can be found on humans: commensal bacteria, which is helpful or harmless, and pathogenic bacteria, which is hurtful. Next slide, please. Now, there are 2 different ways I could have approached this. I could have used a cultural approach where I would use petri dishes and incubate it, or I could use a genomic approach, which that's what I did. I used a genomic mining software to get a list of all the secondary metabolites that could potentially be produced by a certain strain of bacteria. Next slide, please. The purpose of my specific study is to figure out whether a commensal or a pathogenic strain of E. coli is more likely to produce an antibiotic. My hypothesis states that the pathogenic E. coli strains will yield more numbers and types of biosynthetic genes than their commensal counterparts. Next slide, please. The first thing I did was found what strain I wanted to use and found the ref sequence number. Later on, I inputted this into anti-SMASH, and finally, I got a result that looked like this. Next slide, please. Next slide, please. That looked like this. It was divided into 4 different regions to show the different secondary metabolites that could potentially be produced. Next slide, please. Now, this is a table of all the different strains that I tested and the different secondary metabolites that were either present or absent in certain strain of bacteria. Next slide, please. I found that NRPS and thiopeptide were found in all 15 of the strains that I tested, but they're already being used in pharmacology, so this wouldn't help that much. Bacteriocin was one of my most interesting finds because as of right now, it's being used to help prevent the spoilage of food. But after I started my research, an article was published showing that bacteriocin could have an alternative therapeutic effect. This means that it would act differently than the typical antibiotic. It also showed that what's so interesting is that the bacteriocin would kill strains of itself that are similar or identical rather than the parent cell of the bacteria. And bacteriocin is very toxic to bacteria, but not at all to humans. So if and when it is ingested as medication, we wouldn't have to worry about the side effects of it. So for those two reasons and its alternative therapeutic potential, this would be a very important find. Finally, laterine and aeropoline were found in 8 of my pathogenic strains of bacteria, but they did not necessarily have pharmaceutical potential. Next slide, please. So in conclusion, my hypothesis stands because my pathogenic strains had either 4 or 5 secondary metabolites, whereas my commensal strains only had a total of 2. Next slide, please. Some future research that I would hope to conduct would consist of a second phase of my study where I would use a cultural approach. The advantage of having done the genomic approach first is I already know what type of bacteria I would like to use in order to get an antibiotic to be produced. I would combine the commensal bacteria with the soil bacteria in the hopes that the soil bacteria would create an antibiotic and kill off the commensal bacteria. This is where it ties back to the Tiny Earth Initiative to unearth antibiotics from the soil. Next slide, please. I would like to thank Dr. Garcia for mentoring me and being so generous with her time and knowledge, and thank Ms. Thompson for guiding me in biology and research methods.
Awesome job. Thank you so much, Chloe. Our next speaker, we don't have any time for questions. Our next speaker is going to be Kaylin. If you have any questions for Chloe, what you can do is you can send her a message in the Zoom and she can respond directly to you. So Kaylin, are you with us? Yes, I see you.
Hi, I'm here.
Great. So Kaylin, if you want to go ahead and introduce yourself and take it away.
Okay, my name is Kaylyn Hewitt. I am from the University of Wisconsin-Madison, and I was in the Tiny Earth Lab with Dr. Piltorak. My research focused on antibiotic production in fertilized and unfertilized soil. And so as many of us know, we are in the midst of an antibiotic crisis. And what this means is, well, there are 3 main factors that go into why this is a crisis. The first being the problem of antibiotic resistance. This has increased a lot over the past few decades, and it results from, like Chloe said, overprescription of antibiotics and not taking the full dosage. And so these can cause the problem of antibiotic resistance. Also, to cause this crisis, we have the decline in antibiotic discovery. So if you could scroll over to the abstract section to the left, From around 1950 to 1960, we had what was known as the Golden Age, where there was a huge increase in antibiotic discovery. Unfortunately, since then, this has declined a lot and we don't have the same rate of discovery as we have. And so that, coupled with the rise in antibiotic resistance, has caused an issue for the medical field. And that graph on the right just shows the increase in antibiotic resistance for 3 of the main resistant strains of bacteria. And so the third factor in this crisis is that of fertilizer and soil. And these are connected, as we know, through research by the Tiny Earth Lab and other labs, that soil hold lots of bacteria, and we can create antibiotic compounds from these bacteria. And so the rise in fertilizer over the past few years and the likelihood of its continuation into the future has the potential to pose a threat to the production of antibiotics from soil. And that's what my research sought to look into. So we hypothesized that fertilizer would increase the number of colonies from soil bacteria that would produce antibiotic compounds. So the way that we went about doing this was through a series of steps. We obtained soil from Madison, Wisconsin, and we performed a serial dilution to get the soil into a form that we could plate and see individual colonies. We also performed spread patch plating with both the soil bacteria and escape relatives. The escape safe relatives are related to some of the most resistant bacteria, and the 2 that we use specifically are E. coli and Acinetobacter bailey. And these were used just to mimic the most resistant bacteria, just in a safer form. We also wanted to perform PCR and gel electrophoresis to help with identification and providing other background information on the bacteria. And finally, we used Fisher's exact test to compare the proportions of producer and non-producers for fertilized and unfertilized soil. And the picture on the right shows an example of a plate with a zone of inhibition. And this zone of inhibition is kind of our indicator that the colony was producing an antibiotic compound. And so the blue arrow is just pointing out the zone of inhibition. And this is what we would look for to determine whether or not that soil sample was producing antibiotic compounds. And so for our results, we found that unfertilized soil samples produced more zones of inhibition than the fertilized soil samples did. And so this translated to about 11.1% of the unfertilized soil colonies and 3.6% of fertilized samples. And the chart to the right just is a visual representation of our findings. And we can see that the proportion of unfertilized colonies that produced antibiotic compounds was higher than that of the fertilized soil. And so we can see that the application of fertilizer did not appear to increase the number of isolates that produced antibiotic compounds. When performing Fisher's exact test for these proportions, we received a p-value of 0.041. And when we compared this to the confidence interval of 0.05, we can see that our results were significant. And though PCR and gel electrophoresis were intended to help us with our conclusion, we did not receive any conclusive evidence from these, and therefore they were not included in our conclusion. And so if we scroll over to the conclusion section, we can see that the fertilized samples showed a reduced number of colonies that produced antibiotic compounds when compared to the unfertilized soil samples. And so though the findings from the study don't support our initial hypothesis, we still did receive a significant result, and that's shown by the p-value of 0.041. So although we do have a significant result, there are some limitations that mainly lie in the small sample size and small sample radius. And so these would be increased in the future to provide a better study. But overall, when we compare the dramatic increase in worldwide fertilizer usage with the surge in prevalence of antibiotic resistance, we can see a correlation between the two, and this has implications for the future of antibiotic discovery.
Awesome. Thank you so much. Kaylin, we are out of time for questions. If you have questions for Kaylin, you can go ahead and message her and she can respond. Jacob et al. Jacob, are you presenting your presentation today, or is Anna also with you, and Sally?
I'll be presenting it today.
Okay, Jacob. Great. So if you want to go ahead and introduce yourself, tell us a little about you and your group.
I'm Jacob Christensen. Me, Anna, and Sally are all part of the APEX program at Waukee High School. It's a program, it stands for Aspiring Professional Experience, where we work on value-added projects and try to get professional experience and do real-world kind of activities. So we chose to do the Tiny Earth project. So the antibiotic resistance crisis is obviously a big deal. We've heard about it from the two previous presenters. So in our way to combat it, we collected soil samples. Each one of us collected a different one from various locations around our campus. And then we dissolved the soil in water and performed serial dilutions to separate out the colonies so that we could each pick 20. So each of us had 20 separate bacterial colonies that we tested against safe escapes. So our next step, we used the pick and patch method, where we took those 20 colonies and tested them against some of the safe escape relatives, which are relatives of the escape pathogens, which are some of the most likely pathogens to develop antibiotic resistance and the most likely to cause hospital-acquired infections. So we test against the non-pathogenic relatives of those. You can see in our results there that we found Anna in her soil sample found two. She found one. Her number 10 colony had a ring of inhibition against Staphylococcus epidermis. And then on mine, my 8 and 14 colonies showed an inhibition ring against Acinetobacter bailii and Staphylococcus epidermis. Staphylococcus epidermis is the safe relative of Staphylococcus aureus, and Acinetobacter is the safe relative of Acinetobacter baumannii. So they're both, so hopefully that shows that they'll be able to produce antibiotics against those. So those rings of inhibition prove that they might be producing antibiotics that could be useful. Then we did PCR, amplified the 16S gene. We showed that in the gel electrophoresis. You can see the bands there, that the 16S gene was amplified. We haven't gone to do the sequencing yet, just due to problems with online school in general, just 2020 things. But that was as far as we got. We have those 3 different colonies showing antibiotic production and inhibition. Just thank you for thanks to the Tiny Earth Project for the opportunity to do this. We also want to thank the Iowa Science Foundation, who gave us a grant to help us be able to fund this project, and also like to thank our instructor Dr. Holly Showalter, who really helped us out with this. It was really the first time she'd been a Tiny Earth instructor due to being trained in January, kind of before this whole thing started. So really the first time our program had gotten the chance to do this. And it was an amazing experience, and we'll hope to be able to continue to research on this and learn more about those colonies we found.
Great, thank you so much. So our next speaker is going to be, Merritt. Merritt, are you with us?
Yes. Hello.
Great, thank you, Merritt.
Hello. My name is Merritt Simmons, and I'm from Beloit College. I'm having some issues with my Wi-Fi, so bear with me as I'm not able to use my video today. I will be presenting on the optimization of natural product extraction methods for the Tiny Earth Chem course. So a new Tiny Earth Chemistry course is being developed by a number of different instructors and researchers with a focus on the extraction and characterization of secondary metabolites from antibiotic-producing bacterial strains. The goal of this project was to find robust methods and strains for this course that would work well in undergraduate teaching labs. The extraction and characterization sequence I was working with involves growing liquid cultures of bacterial strains, extracting natural products through either liquid-liquid or solid phase extraction, and performing disc diffusion, TLC, and TLC bioautography assays on the resulting extract. For the optimization of these methods, I'll start with the strains. I investigated 4 producer strains of interest shown on the bottom left. L. antibioticus, which is sometimes used as an antibiotic producer control in Tiny Earth and produces a pink, orange, or brownish pigment. AH48 from the Tiny Earth Chem Hub, which is a Bacillus amyloliquefaciens and synthesizes an antibiotic protein. 3672 from the Tiny Earth Chem Hub, which belongs to the genus Streptomyces and produces a purple pigment on PDA. And lastly, S. chartreusης, which produces pigmented chartreusine antibiotic. I mainly worked with Tech AH48 and 3672 as I found both to produce reliable results. The L. antibioticus I was working with was finicky, and the S. chartreusης is a strain our lab doesn't yet have, but we did a lot of research about it and believe it would work well with these methods in a teaching lab because the known pigment it produces could be purchased and used as a control. So moving on to figure 1, as I mentioned, strain 3672 produces a pigment on PDA. Figure 1 shows that an extract of this strain grown in LB on the left yields 10 milligrams per 150 mils liquid culture, while the same strain grown in PDB on the right yields over twice as much extract with a much darker color. So we can see this strain is producing natural products in PDB that it's not in LB. I also determined that 1 week growth is satisfactory for both of the strains I was working with. The mass of extract obtained leveled off at just before a week of liquid culture growth. So figure 2 shows 3 TLC plates of strain 3672 grown in PDB comparing extraction methods. Plate 1 is the extract obtained from liquid-liquid extraction. 2 is from solid phase extraction where the liquid culture was grown 1 week, then shaken with solid phase extraction resin for 4 hours. And 3 is from solid phase extraction where the liquid culture was grown with resin for 1 week. If I had more time, I would have liked to repeat this experiment as it was my first time working with the resin and I had some difficulties with it. So I would have liked to perform some additional trials there. And figure 3 shows some work with TLC bioautography. The top is an AH48 extract and the bottom is a 3672 extract. The zones of inhibition on the plates are circled in red as they're a bit difficult to see in those photos. And I found it useful to use a tetracycline disc as a control for comparison, which you can see on those plates. One observation I found really interesting was that for strain 3672 on the bottom there, the pigment started appearing around the zone about a day after the zone appeared. And you can see that pigment on the top of the plate there. So in conclusion, for the strains, Tech AH48 and 3672 were found to produce reliable results. L. antibioticus was inconsistent, so we're moving away from using at least our stocks of it in our lab. And working with a strain that produces a known pigment is useful. Also, 1 week growth was found to be satisfactory for the strains I worked with. For extraction, liquid-liquid extraction was most reliable, while solid phase extraction required less solvent for large liquid cultures, but was much more finicky. And for the assays, a tetracycline disc control was found to be useful in TLC bioautography. And for pigment producing strains, the pigment may appear after zones of inhibition, which can be an interesting observation. And again, if I had more time, I would want to work more with solid phase extraction and perform more trials of that extraction experiment. I've worked on a few projects related to Tiny Earth with Professor Kristen Labby at Beloit, and I'm really grateful to have had the opportunity to conduct research and to see both the lab work and curriculum development side of things. And we would like to thank Catherine Maloney and the Tiny Earth Chemistry course development team for pioneering this work, the Tiny Earth Chem Hub for sharing strains AH48 and 3672, as well as the Beloit College Honors Term Program and Beloit College Chemistry Department for sponsoring this work. Thank you.
Thank you, Merritt. Megan, you are up next. Megan, are you here?
Yes, I am.
Great, thank you. Go ahead and introduce yourself.
Hi, everyone. My name is Megan McGuire. I'm a senior this year at the University of Wisconsin-Green Bay. And my research today is around the antibiotic resistance, just like the other presenters today. And I'm going to be presenting on my research, the antibacterial activity in a cornfield. So like has been said before, antibiotic resistance, it's a global issue due to the overuse and misuse of antibiotics. And the factors of the antibiotic resistance include bacterial infections, treating them and injecting them into livestock. Certain species of bacteria known as the escape pathogens, they're the most deadly bacterial infections, and they're usually nosocomial infections, and treatment of these species of bacteria can be difficult as well as expensive. So in this lab, I used their safe relatives, Staph epidermidis and E. coli. So to begin, I collected a soil sample and plated it to get bacterial isolates. These isolates were picked and patched for a total of 40 isolates on 2 master plates that would be used throughout the duration of the lab. And then the isolates from the master plates were picked and patched again onto 2 separate plates, one containing Staph epidermidis and one containing E. coli. So look for any zones of inhibition. And I found that there were 4 zones of inhibition. So the isolates, those isolates were sampled and sent over to Yale University, where they were able to use blast analysis on the 16S ribosomal RNA gene to get the sequence of the genome of each bacterial species or isolate. So while we were waiting for the results from that, I gram stained the 4 isolates, and the isolates were 6, 15, 14, and 19. And they also underwent organic extractions of the antibiotics using ethyl acetate. So the results from the gram stain, I found that one of the isolates was gram-positive rod-shaped bacilli, and the other 3 isolates were gram-negative rod-shaped bacilli. And this was later confirmed in the Yale blast analysis. The isolate 6, 14, and 15 were all gram-negative. Isolate 6 was Pseudomonas, isolate 14 was Bacillus, and isolate 15 was Myroides marinus. And isolate 19 was inconclusive for the shape because there was an invalid sequence, but it can be assumed that it was the gram-positive rod-shaped bacilli, as the others were gram-negative. And then as for the results of the organic extraction, that was also inconclusive. It just, I missed the last day of lab. I was sick, so I wasn't able to look at the results of that. But yeah, that's it.
Great, thank you so much. Our next speaker is going to be Carolyn. Carolyn, are you with us?
Yes, I am.
Great, Carolyn, if you want to go ahead and introduce yourself.
Yes, absolutely. I'm Carolyn Latour. I am a senior at UWG in Green Bay. I will get started right away. As well as everyone has said, we are doing the Tiny Earth project to hopefully obtain more antibiotic discovery from soils. So I collected my soil sample from a very dense forest near my childhood home. I specifically chose this area because this soil was never subject to any agricultural inputs and it had a diverse growth of trees and other plants. And I was very hopeful that these factors would contribute to my soil sample being very diverse. So then soil dilution was performed on the soil sample to dilute the concentration of bacteria, which allowed for better colony isolation on the agar plates we used. The agar that we used contained cyclohexamide, which inhibited any growth from fungi that was in the soil. We then did picking and patching technique of the unique colonies on the PDA, and then 10% TSA agar plates with cyclo was performed to create master plates of these colonies. We used PDA and TSA to promote antibiotic production. Then these testing on the isolates for antibiotic production was against S. epidermidis, which is a gram positive, and then E. coli, which is gram negative. With this results, 3 isolates showed rings of inhibition, meaning they have antibiotic producing potential. With that, isolate 8 and 16 had antibiotic production against S. epidermidis, and then isolate 14 had antibiotic production against E. coli. These isolates were gram stained. They were subjected to colony PCR to amplify the 16S ribosomal RNA gene, and blast analysis to aid in the identification of the isolates. Isolate 18 is gram positive and is a Streptomyces genus. Isolate 14 is a gram negative Pseudomonas genus. And then isolate 16 is a gram positive Bacillus genus. Different biochemical characterization tests were also performed on these isolates. The results of these tests correlated to the gram staining results and blast analysis that was performed. Some of the significant results that I got was on the catalase test, isolates 8 and 16 showed positive results containing catalase. And then the McConkey agar test, isolate 8 and 16 showed inhibition of growth, which the McConkey agar promotes gram negative growth. And then just some of the significant findings that I had throughout my research was that blast analysis indicated that the genus of isolate 8 was Streptomyces, which in the past has been used to for other antibiotic production. And then, yeah, the Streptomyces species are known to produce antibiotics. I was really grateful to have this opportunity, and I just want to end with thanking Dr. Brian Merkel, my professor, who showed support and his expertise through this research, which made the whole process really, really great. Thank you.
Thank you. Do you have any questions? We have about a minute. Any questions for Carolyn? All right, if you have any questions, you can message her in the private chat feature. Jacob, are you with us? Jacob? Jacob? I'm just scrolling through for participants. does not appear to be with us today. All right, well, that's going to conclude the poster sessions for this session. We would ask that you would come back for the into the main room then. Our next session is going to start at 3 p.m. We have a little break here till 3 p.m. Eastern time for our next keynote address. So if you would come back at 3 o'clock for our next presentation, and I'll be in the main room for our keynote.
Thanks, Eric. Good job, everyone.